Thank you for joining the Once Changing the World, which is India's first future tech meets sustainability podcast. And today I'm delighted and honored to have with me Dr. Alysson Muotri, who's a professor in the Department of Pediatrics and Cellular and Molecular Medicine at the University of California. He's a member of the Stanford Consortium for Regenerative Medicine, founder of Tismoo, the first laboratory in the world dedicated exclusively to personalized medicine for autism spectrum disorder and other autism-related neurological disorders with a genetic origin. Dr. Muotri is studying the brain from an evolutionary and developmental perspective, differentiating stem cells to recreate brain organoids in the controlled setting of a lab. This work has implications for the generation of human disease models by determining the molecular and cellular mechanisms driving neurological complex disorders such as autism. It is also creating opportunities for identifying and testing novel therapeutic approaches. So, Doctor, I really appreciate you taking time and being part of our humble podcast. Why don't we start with a little background, your work, the mini brain or brain organoids that you're working on?
Thank you so much for having me. My pleasure to talk about our work. Yeah, so brain organoids are stem cell-derived tissues that you can create in the lab. So these are artificial tissues that we induce the stem cells to mimic the pathway that the brain does during development. So we are basically replaying neurodevelopment in a dish so we can follow all the stages. The beauty of the process is that the cells are genetically pre-programmed to do that, so they know what to do. It's not that we need to teach them all the steps, how to migrate, how to form the cortex, things like that. They know how to do it. So we just kickstart the process by adding some chemicals to the cells. The cells receive the signaling, and then they start to differentiate or to specialize in the brain tissues. And over the years, we have been optimizing the protocol. So we have these three-dimensional structures that not only resemble the human fetal embryonic brain, but very recently we got to the point where they can generate these microcircuitries that mimics exactly the preterm development in humans.
How cool is this? But I mean, before we get into this topic further, I want to bring the audience. I mean, give them a little understanding of how do you go about building this brain organoids that you're talking about. You're using stem cells with induced pluripotent stem cells. Talk about the process. How does this work? And once you kind of build these mini brains, what are the applications of these brain organoids?
Yeah, so these are great questions. So you can create the brain organoids from human pluripotent stem cells, and there are two flavors from them. One is human embryonic stem cells. These are stem cells that we can isolate directly from the human blastocyst. Most of the time, we acquire those cells through in vitro fertilization clinics who dispose the non-used blastocyst to the science, so we can remove the embryonic cells from them. Another common source is to use, as mentioned, human induced pluripotent stem cells. These are stem cells that we can reprogram from a live person. You and me, I can take like a small piece of your skin, for example, or your blood or your dental pulp, and I can reprogram the cells back to this embryonic-like stage. And from that stage, I can create these brain organoids. So there are many uses for that. I mean, the main reason why we do that is because we don't know how the human brain is formed. And this is because it's very inaccessible. So it happens inside the uterus of a woman. So we don't have non-invasive technologies to visualize how the cells migrate, how the cells start to connect to each other, how the first synapses are formed, why the first neurons fire. So we have really basic fundamental questions about the origins of the human brain that we have no idea. It's a black box. I mean, for years, researchers have been extrapolating the knowledge from a mouse embryo. But the mouse brain is fully formed like in 20 days. And the human brain takes the whole 9 months. And it's still not fully formed when we're born. So it's a long process. And because of these developmental differences, it's quite important to learn from the human material. So the brain organoids allow us to look into the brain formation without having to go inside the uterus. So we can basically recreate the brain outside the uterus and follow all the process. And that's what we're doing. So we are learning how the brain is formed. Now, for example, if I compare this process from a neurotypical person, you and me, compared to someone who has a neurological condition, I can now understand how the neurological conditions appears. And if I understand how that happens, I can perhaps mitigate, or I can block, or I can treat that condition. And by the way, that's the reason why we don't have good treatments for neurological conditions. I mean, you can probably notice that we have good treatments now for heart conditions, for infectious diseases, for blood disorders, because these are accessible tissues. We can study them right away. But the brain remains like a big challenge because of its inaccessibility. So that's why this tool is so powerful. There are many other opportunities that we can use this technology. For example, if we learn how the brain computes, we could perhaps improve artificial intelligence to become more human-like. We can study brain evolution. We can study what's the impact of environmental factors in your brain. We can improve how the brain works, and many things. We can compare our brains with other species. So there's really a vast array of applications that we can do with these brain organoids.
Thank you for sharing that with us, Doctor. It's fascinating. You say that you take the stem cell and then you put in a petri dish and it evolves into a 3D brain organoid. It's fascinating that the stem cell has the data of what a brain could look like or develop into. How fascinating is that? That itself is something, you're unable to fathom, because the brain, like you mentioned, it's a black box, is doing some really, really crazy things. The 80 billion neurons, 100 trillion synapses, the way it fires and wires, it creates all senses, but yet we know so little. But I think there's some crazy scientists out there such as yourself doing some really, really crazy things, when it comes to understanding that black box and possibly creating these applications. Now, you said that from learnings of these the brain organoids, you are able to kind of understand why there is a certain neurological disorder. Now, you guys have been at the forefront of understanding autism. In fact, there was this recent news which I read that through gene therapy, it can reverse effects of Pitt-Hopkins syndrome, which is a form of autism-linked mutation in human brain organoids. Could you kind of elaborate on that? Because there's millions in the world who are suffering from autism. Is this like a ray of hope for possibly those autistic patients?
Yeah, so that's it's one of these results that we are very happy to see. As you mentioned, there are millions of people suffering from autism, and autism is very heterogeneous. So there is different types of autism, and Pitt-Hopkins is a very severe form of autism. So these are people who are very dependent. They don't talk, they don't speak, they don't have mobility. They are in a wheelchair. They have gastrointestinal problems. So their life it's really hard. So if you can find a way to understand how mutations in that gene, it's called TCF4, causes Pitt-Hopkins, maybe we can interfere with that. And that's what we did with the brain organoid. So we created brain organoids from people carrying the TCF4 mutation from Pitt-Hopkins patients. And what we realized is that their brain organoid doesn't form correctly. And because it doesn't form correctly, it has all the consequences of not functioning well. So that was per se a big surprise to see that we can recapitulate so well this neurodevelopmental condition. And then we asked the other thing, what if we put the corrected gene back inside this brain organoid? Will the brain organoid now start to functioning like a neurotypical, like a control brain organoid? And so we use a gene therapy approach, which is to place the correct version of the virus inside, the correct version of the gene inside a virus, and use the virus to deliver the correct form of the gene inside the brain cells, inside the organoids. And they do that very efficiently. So they infect the neurons and they transfer the correct version of the gene. And by doing that, they were able to express the right gene. And suddenly, all the developmental process that we saw for Pitt-Hopkins were gone. They were basically indistinguishable from the controls. So this is very powerful because it brings hope that we can perhaps apply that same technology to humans. And that's exactly where we are now. We are taking this idea, this technology, and starting a clinical trial with real people, so we can infect their brains and see if the same effect that we see in the organoids will help them to overcome the developmental process that causes the condition. So there's major challenges now. A brain organoid is not just a miniaturized version of the brain. So it's a tiny thing. It has like 5 million cells. As you pointed out, the human brain has 86 billion cells. So the orders of magnitude, it's a challenge. So we have to infect way more cells. The organoid doesn't have all the brain structures. The human brain is very complex, have all the different structures, and is fully connected with the rest of the body. So we anticipate that we're going to have to face some of these challenges for this technology to be translated. But nonetheless, we think that by starting this clinical trial, we're already going to learn so much, and we can improve along the way. This is one of the same things as experimental biologists have done. For example, blood transfusion. In the beginning, blood transfusion was not successful. We could not save people by doing that. The same thing for organ transplantation, heart transplant. The first patient died, the second patient died, and then we start learning how to do that better and better, and then we start saving lives. So science has to pass to this experimental phase until we get to a stage where we know what we're doing, we're learning from our mistakes, and we start saving people's life.
Congratulations to you and your team, Doctor, because I guess there's millions in this world who are waiting for what could be like a miracle therapy, for the parents who have their children who are suffering with autism. Could you kind of elaborate on the clinical trial? Could you share updates? And you mentioned that these brain organoids have around 5 million cells. What would it take to create a brain organoid that resembles or the human brain? Because the human brain is extremely complex. It's got these various subdivision parts, and it is responsible for a cognitive output. Is the brain organoid that you built, which with those 5 million cells, does it resemble a certain part of the human brain?
Yeah, so these are all great questions. Let me start with the first one. So the clinical trial, where we are now, so we are taking that same virus that carries the correct form of the gene, and we are testing in monkeys to make sure that there is no toxicity. Because since we are moving to people, now we have an immune system. We have the whole body that's gonna see those viruses. So we need to make sure that there is no side effects. So we do that in monkeys, and we analyze those monkeys, see how they support this virus. Once we see that there is no side effect, then we can move into people. So that process might take a year or so, and then we start the real clinical trial. So that means that we have to manufacture those viruses in a very clean facility, so there is no contaminants in there, and then we will be testing into people. So that's where we are with the clinical trial. So you mentioned about the brain organoid. So the tissue that we have, these 5 million cells, surprisingly, they reproduce really well pieces of the brain. And we have been focusing on the cortex because the cortex is this region in the forebrain that actually helps with cognition in your social life and all the high functioning activities that your body performs. So it's an essential part of the human body and the human brain. That's why we choose to start there. But we have protocols to other pieces as well. So one thing that we are learning how to do is how to create these organoids containing multiple pieces of the brain, so we can increase the complexity. So what are the challenges here? One major challenge that we have is vascularization. As the brain grows and as we add more cells, we need a vascular system. And the vascular system is not part of the brain, so it happens in parallel. So we have to learn how to vascularize the brain organoids so it can grow together. It can grow by sending the nutrients inside the organoids. The reason why right now they are so tiny, they can go up to 0.5 centimeter in diameter, it's because there is no vascularization. So they don't receive the nutrients inside of this sphere, and the cells inside start to become necrotic after a while. So they don't produce more cells to continue to grow. Now, if we add the vascularization, then we can, in theory, grow as much as the size of the human brain. And if we learn all the chemical protocols to create all the different brain regions, in theory, yes, we could recreate the entire brain in a dish. And to be honest, that's the ultimate goal of the research, is to be able to create the entire brain so we can learn from the entire process. As we increase this complexity, there's another challenge that comes together, which is the ethical challenge. You might imagine that at one point, if you are getting closer and closer to the human brain, the functional aspect of the brain is to become self-aware. So they might acquire a level of consciousness. It might not happen now, but in a future, that might be a consequence of this complexity. And that raises some ethical and even moral questions. How do we grow them? Do they have a moral status? How do we discard these entities? These are entities that are not in the philosophy books. I mean, I asked my philosophers and colleagues about that, and they said, well, we never studied that because it doesn't exist in our books. So this is a new class of being that we need to learn how to deal with them. So and that's why I like to grow the sciences together with the bioethicists. So we are always in constant check of this technology and making sure that we are discussing this with the society, so there is no pushback on this technology. Because at the end of the day, I mean, the goal is really to save lives, and there are millions of people suffering from neurological conditions. So that's where we are now.
Right. You mentioned about the ethical and moral implications which could arise, with, these brain organoids becoming self-aware. But if you go to see, I mean, currently, I think we are pushing the boundaries of what we termed impossible, with the field of artificial intelligence. We are working on creating self-aware robots and self-aware machines. Possibly maybe in the next couple of decades, we might be having machines that could have maybe not just think like us, but also possibly be conscious entities. There are already teams around the world who are working on xenobots, and there's some crazy innovation. I guess instead of curbing innovation, we need to find a common ground or involve people from around the world, so we create a conversation on how do we approach this these boundaries or borders that we are pushing in a very collaborative way. I think there needs to be a larger conversation, rather than what's been happening is that most of the innovation is happening from US because I think it's the mecca of innovation in science. I mean, but I think the things are changing very, very fast. There's some insane innovation growth happening in China and so in India. So in this world which is growing rapidly, there is so much downsides of our health. We are questioning on, can we reverse age? These are deep questions, and instead of shying away from them or having these religious blinders, we need to push beyond, because it's an eventuality that in the next few decades we will be getting into the future where we'll have tools in our hand and resources which could give us the potential to possibly reverse age, reverse these diseases, and possibly create, these conscious entities. Yes, so I think we need to have a collaborative, concerted conversation towards this future. Now, for these at least for understanding of brain, we have devices such as EEG, EMGs to kind of understand the structural and functional capabilities of a brain. How does it work with a brain organoid which is so minuscule, which has got only 5 million cells? And you also mentioned in the course of conversation, it's creating these microcircuitry. How do you kind of understand what's happening over there?
Yeah, so I mean, this is a challenge. While we do have good tools to analyze how the brain works, you mentioned EEG, MRI, things like that. So the brain organoids is really tiny. So we also have, which I think is an opportunity, but also a challenge, which is to develop tools to analyze these brain organoids. So we have to rely on tiny microelectrodes that we can place on the outside of these organoids and record from them, or to go and use probes that can go deep inside of the tissue of the organoid, so we can record from those neurons and start learning. So we have to develop the technology as we go. So together, this is absolutely essential. So sometimes because the organoids are so tiny, we cannot use the tools that are available. So we have to step back and really design a tool so we can use the tool with the organoid. We do that all the time, and I'm so glad that now we have like bioprinters. We have things that really help us to customize all this technology for the brain organoids. So that's it's, as you mentioned, things are moving really fast. And it's a really good moment now that we have the different technologies that are converging on something. And the other thing is, even on biology, we have genome editing. We have CRISPRs now that can help us to manipulate the genome and manipulate the genome inside the organoids, not only for gene therapy, but also to understand how the brain is wired, so we can learn more about ourselves. And this is just because we are in a moment in time where all the science is converging. So we have breakthroughs on stem cell biology, breakthroughs on genomics, breakthroughs on biotechnology that allows us to do that.
Some really exciting space. Now you mentioned that that you're working on this gene therapy. I know it's not right of me to ask you, I mean, the timelines, but if you had to kind of give a timeline of bringing this to patients, what would be the timelines, and also talk about the subject of autism, because it's been linked to the gut, and fecal treatments has proved beneficial, but the research was done in a very small number of children. So share your views on the fecal treatment, fecal transplant. Does that work? And as the founder of Tismoo, which is the first laboratory in the world dedicated exclusively to personalized medicine for autism, maybe talk about the Tismoo organization also.
So let me start with the fecal transplant. So there is this idea that your gut-brain axis is involved in autism. The scientific evidence is not strong. Most likely, the same genes that are affecting your brain is affecting your gut. So I think what's happening is the brain is controlling the gut, and the gut is not functioning well because, there is these mutations related to autism. But some people, they think otherwise. They think that is the gut that's influencing your brain. But again, the scientific evidence for that is not a lot. And it's weak, to be honest. Most of that it was done in mouse. So, but even though, it's a hot field, and people are thinking, well, if you fix the gut bacteria or the microbiome inside the gut, it might help the brain to functioning well, which is a crazy idea. But people actually took that to clinical trials. The problem with that is that sometimes, you're manipulating bacteria, and there has been death of patients on this clinical trial. So this is not without a risk. There is a high risk that you are hurting the person by doing that. And that's why I think are slowing down on that scenario. I think it's still worth to pay attention, but again, I think we need more science behind it to support those ideas. And I think it's not coming from the mouse; it has to come from human subjects. Having said that, I think the mainstream idea is that still your brain is affected by the mutations, and that's what our work with brain organoids is clearly showing. Because of those mutations, the brain's not functioning well and might affect all the other tissues in your body. So Tismoo was born with this idea to start linking the genetic variants for each individual, because each individual carries private mutations, and try to understand, using the brain organoids as an avatar to those individuals, how those genetic alterations actually changes how the brain functions, and then uses that to personalize the treatment. So what if we now find small molecules or even repurposed drugs that could help us to mitigate the effects of these mutations? So that's why we decided to make Tismoo really focus on autism, because it's so easy to get lost on so many syndromes that we decided to focus on autism. So we are really expert on that, because we understand what are the genes implicated, and by using this technology, we can understand how they affect neurodevelopment and how to mitigate that using a drug treatment. In the future, gene therapy is our idea as well. So we usually look for drugs that are already FDA approved, so we don't have to wait. So if you find a drug that can be repurposed, you can use right away. You can even use as a compassion treatment, so the doctor can prescribe. And indeed, we have in Tismoo several cases where we find the mutation, and there is already drugs available for treatment of those kids. So we discuss with the doctors, and we say, because of this mutation, there is a drug that actually affects that. And we have cases that was incredible. Untractable seizures in some kids. We do the sequencing, we find the gene, and then we find the medicine that can actually compensate the deficit of that gene. And then, boom, seizures are gone. So it's a major milestone for that patient, just because they did a genome sequencing, something that's so simple as that. And if you have the trained eyes, you can look at the mutations and propose a treatment. It doesn't happen in all the cases. There are many cases where there's nothing we can do, but eventually we do find cases where the treatment is already available. So a gene therapy is the dream if you have a single gene that is mutated, and you can replace the gene by adding the right copy, as we did for Pitt-Hopkins. So that's going to be case by case, because it depends on the gene. Some genes are too big, you cannot fit inside of tiny virus. So for those cases, we have to think differently. We have to use something else. But in the cases where the gene is small, such as the TCF4 that can fit inside the virus, the gene therapy is perhaps the best way to go, because it acts on the root of the problem. So if you fix that, you fix all the downstream consequences of the molecular pathway and the cellular abnormalities, and then the circuitry abnormalities that we have on these brains. So for the Pitt-Hopkins, we really are pushing hard on that, and we are fortunate to team up with a company that has the resources to do all the toxicology testing really fast. So we are anticipating starting clinical trials in 2 or 3 years. And then if the clinical trials are going well, we just increase the number of patients to gain confidence that this treatment is working, and then we can start to commercialize. So I'm assuming that we're going to see that in our lifetime. So if you have a kid with Pitt-Hopkins now, you likely benefit from a gene therapy approach if everything goes well.
I really hope so, and we wish you and the team the very best. Doctor, would you like to share any updates on the brain organoids? Is it being leveraged for drug discovery or other neurological conditions?
Yeah, we are now just my lab, and then there are other labs also working with other conditions. But just my lab, we have, I think, about 25 conditions. These are genetic conditions where we know that the genes is affected, that we are modeling on these brain organoids, leveraging the brain organoid technology. So these are some of quite rare neurological syndromes. There is no even name for these conditions yet. We're just discovering the genes as we go. And there are others that are more well-known. Rett syndrome is another one that we're working hard on it. CDKL5 syndrome is another one. And so for each one of them, I mean, we are in different stages. Some of them we are still characterizing the organoids. Some of them we are already finding like if the gene therapy is working or not. And some of them we decided to go through a pharmacological treatment, so we're screening drugs to see if they can rescue the phenotypes. So depending on the condition, we are at different stages. So Pitt-Hopkins, as you see, is one that we are already on a almost like into a clinical stage.
You mentioned in the course of the conversation that this could possibly help in developing, artificial general intelligence. Maybe share your views on that, and what's the future roadmap? What comes next?
Yeah. So one thing that we thought about, and there are lots of colleagues who also think about that, is that there is some kind of a learning from the human brain that is very intuitive. This is something that you, as a newborn, nobody teaches you, but you start learning right away. So we noticed that, and we don't have a neural basis for that, meaning that nobody understands how the human brain is able to learn by just observing or trying and error. That's how you learn how to walk, for example. So how the brain does that? So if we understand how the brain does that, if we understand how the brain learns, we can apply that to artificial intelligence. Right now, artificial intelligence uses a very rigid network for learning, meaning that this network cannot, for example, generalize. So artificial intelligence relies on a specific learning of a specific subject. For example, how to distinguish cars in driverless cars from people. So you have to train the machine to do that. And once the machine is fully trained, then it can distinguish cars from people. But now, if you add like something else, like a bicycle, it cannot apply to a bicycle. It has to learn again. And the human brain does not do that. The human brain learns everything at the same time. So the idea is, if we leverage the knowledge of how the brain is wired to create that type of learning, maybe we can adapt artificial intelligence to be more human brain-like. So that would be like a huge step on artificial intelligence, not only for the level of learning, but energy efficiency. Our brain is highly efficient. We don't even know how it's so efficient, but artificial intelligence is not. It requires tons of energy to do all this learning, tons of time. Something that the human brain can learn in a couple of seconds just by observation. Artificial intelligence will take several rounds of learning and tons of energy to acquire that. So how that's possible? If we learn how to do that, we can create artificial intelligence that will use a human-like intuition for learning. That will be a game changer for so many applications.
Thank you, Professor. Really appreciate you taking time, being part of a podcast. Wish you and the team the very best. I think we're in the cusp of creating some greatness. I think the entire world is going to change in the next few years, and it's largely because of professors, scientists, and thinkers who are pushing the boundaries of what was possible. And we living in exciting times. So thank you for doing what you're doing. Really appreciate you taking time and being part of the podcast. To my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye bye. Thank you, Professor. Really appreciate this.
Thank you so much. My pleasure.